Yeah, I'm very excited to talk with you all today about some of the ongoing research out of our laboratory. And once again, thanks for inviting me to give a talk with you guys today. So let's just make sure the slides advance. And they are. Perfect. So also, this is the title, but I also should have put in here germline genetics and genomics, because I'm not going to be talking really at all about somatic or cancer genetics and genomics. I do not have any disclosures. So the goal of this talk is really to address areas of uncertainty within germline genetics genomics and sort of how current technologies are really enabling us to address these areas of uncertainty and sort of move past them. And I'm going to start off today with a case presentation. This is actually a patient I saw recently in the cardiovascular genetics clinic here at the University of Washington. So this is a patient who presented initially with shortness of breath, and she presented to the emergency department. Although she was eventually diagnosed with influenza, they nonetheless got an EKG on her at the time of her presentation there. This EKG ended up showing a type 1 Brugada pattern, and she was subsequently referred to cardiovascular genetics for further evaluation. Her past medical history was notable for a syncopal event in her 30s while driving. An EKG done afterwards was abnormal, and her baseline EKG did not show Brugada pattern. Her family history does have this individual with a sudden death in the 40s while playing basketball, as well as a brother with syncope. So just based on this clinical presentation, you can give this individual a diagnosis of probable or definitive Brugada syndrome on the clinical criteria grounds at all. But obviously, really what we're trying to do right now is not just understand what the actual diagnosis is, but actually the genetic cause of her diagnosis. And really, I think this goes back to the concept that for this individual, and specifically for her family, understanding the molecular causes of her diagnosis can be essential because her relatives may have inherited that genetic variant, but we may not be able to diagnose them with Brugada syndrome just based on ECG testing in and of itself. So that genetics is really going to play an essential role in terms of their care. Okay, so before I go over her genetic testing results, I want to discuss how we choose which genes to test for. So confusingly, genetic alterations over 21 genes have been reported to be caused Brugada syndrome. However, many of these disease gene associations have minimal evidence of actually being associated with Brugada syndrome, but are nonetheless being tested clinically. So you can ask yourself which of these associations is actually real and should be tested. So to understand which genes are truly associated with these different diseases, the ClinGen, or Clinical Genomic Consortium, a resource consortium, has put together lists of understanding the evidence base for different disease gene classifications. And these classifications really have markedly improved the standardization of clinical genetic testing around the country, and these efforts are really being enabled us to move past sort of these areas of uncertain genes into ones for which we have much more confidence in when we're actually doing clinical genetics. So for example, a recent effort focused on Brugada syndrome said that of those 21 genes that have been associated with Brugada syndrome, actually only SCN5A had definitive evidence for actually causing Brugada syndrome. So you're sort of whittling those 21 down to the one which you should actually use for clinical genetic testing. Okay. Another piece of background I want to go into before I discuss her genetic results is sort of the nomenclature we're using. This probably is going to be basic material for a lot of individuals in this audience, but I just want to make sure that everyone's on the same page when I'm sort of going over these variants later on. So oftentimes when we're speaking about germline variants, we split these into sort of common and rare variants based on their prevalence within the population. It's important to note that for pretty much the far majority of both common and rare variants, most of these are not going to cause disease, but are actually sort of benign. However, when we're thinking of medical genetics and specifically rare diseases, let me get my laser pointer here, we're really focusing mostly on these rare variants here. And furthermore, most of these rare variants are also not going to be associated with disease. When we think about rare variants, we will split these into sort of 5 categorizations, and this is based on the 2015 Richards et al. Paper out of the ACMG. And the first 2 major categorizations are variants that are labeled sort of pathogenic or likely pathogenic. And really this is that there's sufficient or strong evidence to state that these variants are actually causative of disease. The second sort of grouping is variants which are classified as likely benign or benign, and this is sort of ones which are sufficient or strong evidence to say that this variant does not cause disease. And then there's this group in the middle called VUSs, which I'm sure many people have heard all about these and sort of the challenges that these pose in terms of not only individuals on the laboratory side, but also on the clinical side, which I'll go over in the next couple of slides. Okay, so VUSs are a major clinical problem. I'm sure many people have seen portions or this exact same slide many times over the last couple of years, and it really is, this is an issue. We see these quite frequently, and as more people undergo genetic testing, we're identifying more VUSs. The problem is that most of these VUSs will likely be reclassified as benign or likely benign, but you don't know which ones ahead of time. And so you can't say definitively which way they're going to be. And as a result, we do not use VUSs when it comes to actually counseling family members to inform their disease risk per se, because this inherently is uncertain, and that's sort of the nature of it being a VUS in and of itself. Okay. So with that background, we get back to our patient. So we did genetic testing on her, and we identified a VUS in the gene SCN5A. So why is this variant classified as a VUS? So it's classified as a VUS because it is absent from large population-controlled databases. However, it's important to note that current population databases do not adequately reflect genetic diversity in individuals of non-European ancestry, and this individual happens to be of African-American ancestry. Furthermore, this variant is classified as a VUS because it's never been previously seen in a patient with Brugada syndrome. We don't have any clinical information other than this patient. And there's also no existing functional evidence regarding how this variant impacts the function of SCN5A. Okay, so the next logical question is, how do we resolve VUSs like this one? So first, large efforts are currently underway to make and include more diverse populations in our reference maps of genetic variation. Second, I think it's a critical point, which I'll emphasize a little bit later, is data sharing is essential for us to ensure that when we recognize a patient with a variant, we actually find other individuals who may have that exact same variant to sort of connect the dots, as it said, in terms of individuals who may have this condition. And finally, it's really paramount that we leverage these emerging high-throughput functional assays to inform variant interpretations, not just on the research side, but also on the clinical side. And now I'll go over that in the next couple of slides, how we're approaching that specific problem. And really, I think it's probably obvious to this room, but the University of Washington has really been a leader in this last point in terms of functional variants. We have some of the people who really started creating this concept of how do we actually identify massively parallel variant effects. And it's really sort of paramount for us to leverage this local resource for the patients that we're seeing here, as well as patients around the world who are getting VUSs like this. Okay, so now to this, we can ask the simple question is, can we solve this particular patient's variant through these high throughput functional assays? So thankfully, Andrew Glazer and Dan Roden at Vanderbilt had actually recently established a well-validated high throughput patch clamp assay to identify variants that disrupt SCN5A function. This is just an example here where essentially you have the wild type SCN5A protein, which has normal activity. And then if you have these variants that are pathogenic, they essentially disrupt activity, and which results in this sort of flatlining of current in this patch clamp assay. So they tested the function of this patient's variant in this high throughput assay and demonstrated that this variant completely ablates the ability of SCN5A to work in this particular assay, which has been subsequently well validated. So what that means is that we can now use this functional data along with other ACMG criteria to say that actually this variant is not a VUS, but actually is indeed a likely pathogenic variant, enabling us to do subsequent cascade testing with this family. However, this is just one patient. So we wanted to know, can we do this, a similar approach to resolve VUSs for all the patients that we're actually seeing here at the UW clinic? So how did we approach this? So to address this, we founded actually the Bronfman Beatty Institute Clinical Variant Database. So this is founded last year, which is a collaboration between the University of Washington and the Fred Hutch Clinics, co-led by myself, Fuki, and Marianne. And really this has been enabled with funding through the BBI, as well as the tireless efforts of Andrew Folta and Adriana Cedeno Cortez. So this is a repository of genetic invariants encountered clinically here at the University of Washington and the Fred Hutch. And it's really, the goal is to connect patients with rare diseases to researchers doing high throughput functional studies and developing diagnostics and therapies for rare diseases. So to date, this database includes over 2,700 participants with over 3,100 EHR-linked germline genetic variants, as is shown here. And we're continually adding this at a good pace. This database includes structured data on diverse genetic testing, including sequence-based testing, chromosomal microarrays, karyotypes, repeat expansion testing, you name it. And you can see that this includes data generated across a variety of different clinical labs, as is shown in this chart here. In addition, we are capturing detailed structured data regarding the indication for testing, be it the direct indication in terms of the patient's phenotype or family history, as well as within this individual structured features such as patient's history of specific cancer types, either be it at the time of testing or prior to testing. So using this data, we can empirically see that actually around 50% of the variants that we are identifying in the clinics here at the University of Washington Fred Hutch are indeed VUSs. So this is not just a theoretical research problem, this is actually a practical problem that we're seeing here in our clinics. Furthermore, if you actually break this down by individual genes, so now I'm showing you the contribution of VUSs, pathogenic or likely pathogenic, within our database based on individual genes, you can see that this burden of VUSs definitely varies across different genes, and also this highlights the diversity in terms of the patient population that we're seeing within this cohort. Okay, so what are we actually doing with this database? So the first thing we're doing with this database is we're actually working with ITHS, who is integrating this database into LEAF. And the goal of this is to actually enable cohort discovery using this germline genetic data, as well as information contained within the UW-EHR, so that individuals can develop more streamlined or intuitive clinical trials when working in the University of Washington system. In addition, we're working with Leah, Doug, Brian here, Abby, and Alan, and others to actually leverage this data in addition to the functional data which is being developed here at the University of Washington as well as around the world to actually identify if these VUSs can be reclassified using this functional data, similar to that case I said earlier. So we can now take this patient-level phenotypic information, these patient-level germline genetic information, combine it with this functional data, and actually enable reclassification and subsequent improved potential outcomes in terms of our patients by enabling them to actually have correct genetic diagnoses rather than being in this limbo of a VUS land. Okay, so back to the original sort of objectives of this lecture, I hope that you recognize that there are definitely some areas of uncertainty in clinical genetics and some steps that we and others are really right now trying to take to sort of overcome and resolve these. But it's important to recognize that everything I've talked about so far has largely been focused on a very small portion of the genetic material, specifically 1% of the genome that encodes for genes. In the next part of this talk, I'm actually going to switch gears and start talking about how we can understand the function of the other 99% of the genome. However, to do this, it's really important to understand how the genome is packaged into the nucleus, as this packaging is directly tied to the function of these various genomic elements. So first, I just want to quickly say that the genome does not look like this inside of a cell. So rather, the genome is packaged into chromatin, which is comprised of various proteins such as histones, transcription factors, polymerases, etc., that bind to the DNA in a very specific manner. This architecture of how proteins are actually bound along the genome is essential for creating function for different portions of the genome, and quantifying this protein occupancy has really been essential for us to understand which parts of the genome are involved in different disease processes. So how do we in genetic alterations, these elements can cause many different rare as well as common diseases. So how do we actually identify these regulatory elements? So traditionally, the methodology for doing this is to treat cells with an enzyme that will essentially non-specifically cut the genome where it's accessible or open. So the genome inside of your cell is compacted in a manner so that parts of the genome that are being actively used by the cell are actually quite accessible. So if you were to send an enzyme in there that's non-specifically cutting DNA, it will selectively cut those accessible portions along the genome. You can then isolate these small DNA fragments that originate from these areas of accessibility and sequence them using short-read genome sequencers. When you do this, you get patterns that look like this, and you can do this via a number of different enzymes that will non-specifically cut DNA, such as TN5 in the case of ATAC-seq, DNase1 in the case of DNase-seq, MNase will do this as well. And when you do this, you identify these peaks of accessibility, which correspond to known promoters, enhancers, and other sort of distal regulatory elements along the genome. And this is just showing you that these can be both cell selective as well as universal in terms of their activity across different cell types. If you do this during different cell types or during differentiation, you can see that the amount or the utilization of the non-coding genome massively changes during development. For example, a regulatory cell or a cell can change its regulatory landscape by over 50% during less than 12 or 14 days of differentiation or development. Okay, so this has been sort of the workhorse of chromatin biology for the last decade or so, but really it's still, although it's been quite powerful, it comes with some potential challenges in terms of our ability to understand what's actually going on. And specifically, you can sort of imagine this is our toy example of a chromatin fiber that I'm showing here, but really when you're doing this assay, you're not actually seeing what's going on a chromatin fiber. Instead, you're getting a very narrow view of a specific area because you're only sequencing a small 200, 150 base pair fragment, and it's actually being cut from the surrounding DNA. So you have no idea what's going on that same molecule of DNA when you're doing this. You really can't resolve the actual architecture of this intact fiber. Furthermore, since we're actually cutting these into very small fragments, you can imagine that this is a nightmare when it comes to actually trying to map this to complex regions of the genome, for which mapping short, small DNA fragments is really not advantageous and really not feasible. So what that means is that there's a large portion of our genome for which we really have no information on when it comes to what's going on from a gene regulatory perspective. So how can we overcome these challenges? And I think really the major goal of this was, can we move away from using scissors to cut the genome at sites of accessibility to using a methodology where we're essentially stenciling the DNA while keeping the underlying DNA intact. And so the way that we're actually accomplishing this is using sort of a spray paint is the way, sort of conceptually think about it, but in practicality, what this ends up being is using non-specific adenine methyltransferases. And specifically what these are enzymes that will methylate the DNA at sites where it's accessible rather than cutting it there. And this concept of using methyltransferases to mark genomic features has actually been around for several decades. And this has been done using sequence-specific methyltransferases such as DAM or enzymes that will methylate at specific GC or CG contexts. However, around a decade ago, there was some observation that bacteriophages produce non-specific adenine methyltransferases. And so really the question that we had was, can we leverage this sort of bacteriophage enzyme for this chromatin perspectives? And one of the benefits of these is that since they're non-specific, it gives us clarity in terms of the chromatin architecture at 59% of the genome, because 59% of the genome are AT bases. And M6A is essentially a non-native mark in humans. So we know if we see a modification on a piece of DNA, we caused it to be there. It wasn't there endogenously that the cell had it naturally at that spot. So how does this protocol work? Essentially, it's very similar to prior chromatin accessibility, but rather than treating cells with an enzyme that cuts, you essentially take your nuclei and treat it with this enzyme that's going to be methylating. We then remove all the proteins after we've done this methylation step, share our DNA around into 16 to 21 kb long fibers, and then we're actually sequencing this with these sort of single molecule long read sequencers. For this specific application, we're going to be using PacBio Hi-Fi sequencing for it, which has a sequencing accuracy of greater than 99.9% and gives us this read length sort of in this distribution right here. One thing to note about sort of the single molecule sequencers is that in addition to the sequence information, you're also often able to capture methylation information at the exact same time on that fiber. In the case of PacBio sequencing, the way that actually works is that when the polymerase, which is being used for PacBio sequencing, encounters a methylated adenine, it actually causes that polymerase to pause a little bit or stall at that site. And that stall or that pause between the incorporation of neighboring bases can be directly read during the sequencing process and used to interpret or intuit where adenine methylation is occurring on an individual fiber basis. And one of the things about PacBio sequencing is it actually is resequencing each piece of DNA multiple times in the process of this through a process called circular consensus sequencing. So in that process of resequencing, you end up getting actually pretty high accuracy with calling individual methylations because you don't have sort of one hit on goal, you end up getting 3 or 4 or 5 hits on goal to be able to sort of average those together and say that yes, this truly was significantly methylated at that site on a per molecule basis. So what does this data look like? So traditionally you imagine you're thinking of chromatin accessibility looking like this, where it's a bulk, either this is pseudocells or it's a bulk tissue. But for this, what we're seeing is on a per molecule basis. So each gray bar here is an individual DNA molecule within the population of cells. So if you imagine you started off this experiment with 100,000 cells, this is a diploid cell line, you would have 200,000 possible molecules of DNA that could map to this area. I'm now showing you 15 of those molecules that are mapping to it here. And on each of those gray bars, if you see a purple dash, that means that there is an M6A methylation that we caused to be there. And what you're seeing is that the predominant signal from this is essentially nucleosomes. These are nucleosome footprints that are around 148 bases in length that are spaced throughout this area with these short internucleosomal linker regions that you're frequently seeing. In addition, you're seeing these sites of chromatin accessibility on a per molecule basis that are corresponding to known promoters as well as distal regulatory elements. What's quite striking once you start looking at chromatin on a per molecule basis, not on a per cell basis, but on a per molecule now, is the amount of heterogeneity that actually exists. So if you look at this particular promoter, you can see that on a per molecule basis, there's some where essentially it's closed down entirely. In some molecules where it's quite accessible, and there's also accessibility to this neighboring element. And this is important because this is the actual chromatin that cell is seeing. This is not sort of heterogeneity in terms of experimental methods, this is true biological heterogeneity. So when we're making our models of what is going on from gene regulation, it's important to say that this is not what the promoter looks like in every single cell or on every single molecule within a cell, but really has this immense amount of heterogeneity there. Furthermore, because we have a base resolution of 59% of the bases, which are AT rich, or which are AT base pairs, we can not only see these sort of sites of chromatin accessibility, but if you actually zoom into them, you can appreciate individual transcription factor footprints. For example, this is a known CTCF binding element, and if you look on a per molecule basis, on these top dozen or so fibers, you're appreciating a footprint that's perfectly overlapping the CTCF element, versus in these bottom 3 fibers, that site where the CTCF should be bound is actually being methylated, indicating that although that site's accessible, it's actually not bound on that single molecule of DNA. Okay, so not only are we having this single molecule data, but as I mentioned, this is long read data. So one of the benefits of long read sequencing, as I'm sure many of you guys are aware of, is its ability to do haplotype phasing because you have these long reads that are often capturing multiple heterozygote variants that could be on the order of several kb apart. So what that means is you can go from bulk data sets that say show you chromatin accessibility that looks like this. And so in this situation, this is a data set of the exist promoter, which is a gene that is known to be selectively active on the inactive X chromosome. And this is actually fiber-seq data we did from a cell type for which only one of the 2 haplotypes is actually turned on. And now if you look at this on a per molecule basis, you can actually see that on the inactive chromosome, you're selectively seeing activation or activity at this promoter, versus this upstream CTCF element appears to actually escape X chromosome, or sort of it appears to be active on both the inactive as well as the active copy of the X chromosome. So I've given you the ability to identify on a per regulatory element basis which elements are actually being haplotype specific and also which elements are potentially escaping or doing odd parts when it comes to the X chromosome. And notably, around 80% of our fibers can be actually haplotype phased using this process. So what that means is that we can go from sort of a vision that looks like this now into one that actually looks like this using this sort of methyltransferase approach. So now back to one of the questions which I was asking earlier, which is what are we missing? Are we missing in terms of our understanding of the actual chromatin accessibility and what are regulatory elements across the genome? So one notable area that is been sort of a huge area of human genome evolution is actually segmental duplications. These comprise around 7% of the human genome and are cause of actually many rare human diseases, which are caused by genetic variants within segmental duplications. However, as you can imagine, based on everything I've told you before, these are quite challenging to study using short-read approaches and specifically short-read chromatin approaches. This is a particular example of that. This is the Notch2NL locus. This is a locus that this is involved in 4 separate Mendelian disorders. Yet if you were to look at the short-read chromatin accessibility, you would have no idea what's actually going on in terms of the gene regulatory architecture of this area because it's quite complex and quite challenging to map the reads to. So how do we resolve this? And first of all, can FiberSeq even resolve this type of things? So the first way we want to approach this was, can we actually call essentially bulk chromatin accessibility using our single molecule approach? And to do this, the way we approach this was using a machine learning approach where essentially we take these methylations on a per molecule basis and essentially use a machine learning algorithm to categorize them as to whether or not those methylations constitute an accessible chromatin patch or a simple internucleosomal linker region. In the next couple of slides, I'm just going to be using colors to denote this. And this is using a semi-supervised machine learning approach for this. So this is data where I'm looking at individual fibers, and you can just see that you're seeing these sort of preponderances of red at certain locations, and then a sea of sort of lavender in the background here. And you can add up the red using approaches where you're sort of summing the FDRs for each individual call, and you end up getting sort of peak profiles that look like this. So these are sort of peaks of accessibility, which are called using this single molecule data. This is in a sort of standard euchromatic portion of the genome that's very easy to map to using short-read data. So you can actually compare this chromatin accessibility that we're deriving from our single molecule data to the bulk data, and you can see that it matches up very nicely. And where all these areas which we're calling accessible are largely areas that are being called accessible with the short-read data, with the notable exception being this one right here. And if we actually look at the sequence of this is actually falls within a retrotransposon that just essentially would not have been mapped to using short-read data. So now we can go back to this locus I showed you earlier. So does this region actually contain chromatin accessibility? So using our FiberSeq data, we can see that actually the Notch2NL promoter is actually quite accessible in the cell type, and also pull out these putative distal regulatory elements that are also contained within this Notch2NL locus. So yes, we actually can now map chromatin accessibility across these quite challenging to map areas of the genome because we have these highly accurate 10, 20 kb long reads that are able to uniquely map to these areas. Okay. If we do this, what we identify is that for a single cell type, 10% of the regulatory elements in that cell type are completely novel, have never been seen before if you look at all of the ENCODE data that's been sort of merged across hundreds upon hundreds of cell types. So I mean that there's a huge amount of gene regulatory elements out there which have just not been studied and are sort of ripe for future discovery. It's important to note that these elements, as you would expect, are really enriched for some of the fastest evolving portions of the human genome, including these areas which are sort of enriched within segmental duplications, retrotransposons, and other mobile elements, which we know are playing a critical role in modulating how gene regulatory activity is unique in humans. But everything I've showed you so far is using HG38. So I'm sure you guys have seen that over the last year, HG38 is imperfect. And there's better reference genomes that are out there, including the CHM13 T2T genome, which came out last year. And so the question we sort of proposed was, what happens if we now move beyond HG38 and go into sort of the CHM13 genome? What are we able to uncover that we couldn't uncover just by using HG38 as a reference? As a little background, with the new T2T genome that came out of CHM13 cells, this was led by Adam Philippi, Karen Miga, and Evan Eichler's group. There was 220 megabases of brand new material that was added to the reference genome, which is equivalent to the size of chromosome 2. So what we wanted to do is ask the simple question, which if we applied our FiberSeq technology to CHM13 cells, so the same cell line that was used to derive this T2T reference genome, what can we uncover that we just haven't been able to look at previously? The first, sort of the simple answer, is we were able to actually get a complete map of CHM13 chromatin from telomere to telomere, including the chromatin architectures of all the CHM13 telomeres and centromeres. And I'll show you some examples of this in the next couple of slides. For example, this is actually the telomere on chromosome 8 on the P arm of it. And so what you can see here is these are individual molecules of DNA that we're mapping using our FibroSeq. This is just showing you the DNA where I'm just sort of simplifying it just to show you where the molecule is mapping to. And you can appreciate that we're actually looking at the heterogeneity in terms of the actual telomere length on a per molecule basis here. But not only do we have the actual length of the molecule, since this is highly accurate sequencing, we have the underlying sequence of it. So now we can just simply color this molecule as to whether or not that molecule contains the standard typical TTA-GGG repeat, which is sort of the telomere repeat at that site, or contains a non-TTA-GGG repeat, so one of these telomere variant or degenerate repeats. And what you can appreciate is that there's a large amount of degeneracy within telomere sequences, that sort of the textbook of TTA-GGG is your telomere is not necessarily the most accurate thing once you start looking at this. And also that this degenerate sequences are actually relatively conserved, almost forming a barcode that distinguishes each telomere from each other. And you do have some of these fun instances where you actually have where they've been actually disappeared or replaced with the standard TTA-GGG repeat, which likely actually reflects in this situation where this telomere at one point was actually shortened past this variant repeat and then re-extended through the action of TERT here to create this sort of baseline TTA-GGG repeat. But as I said, this is FibroSeq, not just standard PacBio sequencing. So not only do we have the sequence and the length, but we also have the chromatin data. So now you can map on here the actual accessibility. So one thing that's quite important that you see from this is there's very little methylations in here. So what we see is that in general, these telomere repeats are forming what you would expect, telomere caps, which are strongly protected from being methylated, by our enzyme. However, when you do see disruptions in the actual cap architecture, they tend to be localized specifically within these variant repeats. So what you're seeing here is the actual impact of these degenerate repeats on the overlying telomere cap architecture. And just zooming out a little bit from here, this is now showing the telomere repeat and then also the neighboring sort of subtelomeric region. What we were able to appreciate is that on around 85% of the telomeres in CHM13 cells, the actual telomere is bookended by this highly accessible gene regulatory element that's critical for the creation of a protein or of an RNA species called TERA. So you have actually this bookending of an accessible element that's juxtaposed between this highly inaccessible compacted chromatin array on the right-hand side, or on the centromeric side, you could say, of this, and then this transition out into the actual telomere cap architecture. So this is fun. You can actually view this stuff on a per molecule basis and see what's going on in individual molecules of the telomere. So now to jump into the middle of the chromosome and what you can see, the centromere is just as fun. So what you can see is that you can actually map chromatin accessibility throughout the entire centromere. And this is sort of the counterintuitive thing that came up. You usually think of centromeres as heterochromatin. Heterochromatin you sort of equate to being tightly compacted and closed portions of the genome. So if you were just to ignore this thing going on in the center and look at everything over here, what you see is that you have this sort of large area of hyper-CpG methylation corresponding to alpha cytoside array, and you essentially see no accessibility as measured via this FibroSeq assay. And then you get into this little middle area. So this is actually one of the observations that came out of the T2T paper, is that for all the centromeres in the CHM13 genome, there's this hypomethylated region in the middle of the centromere that corresponds to the kinetochore binding region. And one of the striking things, that is actually one of the absolute most accessible portions of the human genome. So if you look at chromatin accessibility within that, it's highly accessible. But it's not accessible in any way that we think about accessibility in terms of the euchromatic portions of the genome. As I showed you in the past, euchromatic portions of the genome, you have a accessible element that's accessible at the same location across multiple fibers. Maybe there's a little bit of heterogeneity. You look inside of here, and it's just a disarray. You essentially have these accessible elements that are located at heterogeneous positions and not a specific location. And if you actually quantify how many accessible elements are actually present on a single molecule of DNA, it actually corresponds to the number of kinetochores which actually are supposed to be bound along an individual molecule of a chromosome. So, yeah. Okay, so That's fun. You can do all this cool stuff with CHM13 cells, and we now have really the tools to look at some of these previously unmapped portions of the genome. But I think that one of the challenges that becomes with this is CHM13 cells are great and all, but that's just one individual out there. Can we move beyond the CHM13 reference genome and sort of start to uncover the sort of full, complete understanding of the epigenome or the chromatin architectures in sort of a more diverse population? And so really we want to know is, can we start building actually a complete human epigenome directly from our data in of itself? So to do this, we actually specifically worked with a particular patient sample to see if we could actually develop, get this methodology to work. And so what we did is we had a patient-derived cell line. We applied FibroSeq to it and actually used this as the baseline, and then supplemented this with ultra-long read nanopore sequencing, as well as short read whole genome sequencing on the actual parental samples. And the goal was to actually leverage some of the newly advanced diploid assemblers that's been produced by Adam Phillippe's group to see if we can actually assemble a de novo genome using this FibroSeq as the baseline or the backbone for it. Using this approach, we were able to actually get 27 of the 46 chromosomes completed telomere to telomere with 0 gaps. So just as out of the box, which is just, I think is not any sort of testament to what we're doing. It's really a testament to what Adam Phillippe's group is able to do in terms of these genome assemblers and really get them to work quite beautifully. But I think that really what this tells you is that using this as a backbone, you're able to not just get your chromatin out of this, your sequence, but also sort of understand the sort of easy and the more challenging portions of the genome simultaneously when you're actually looking at this. And the reason why we actually approached it for this particular patient sample is because they actually have alteration in a gene that's supposed to actually impact the heterochromatin formation in certain areas of the genome. So really understanding these more challenging portions of the genome is really going to be essential for understanding how this particular genetic alteration impacts their disease. Okay, so really just sort of highlighting what I showed you in the last couple of slides, we now have the tools to not only map, but also explore these really previously unmappable regions of the human genome. So now I'm going to sort of bring it back to sort of a question of how can we use this new technology for understanding how genetic variants are impacting chromatin. This actually was one of the earlier observations that we had when looking around the genome, is that when looking at the HTT locus, one of the things that we observed is that actually the area which is the repeat happened to be located in this sort of accessible internucleosomal linker region. And we want to really explore what's actually going on here and whether or not there's something of note that we should be looking into. And specifically, the reason why we were interested in this was because HTT, as many of you guys know, it's a repeat expansion condition caused by CAG repeat expansion. The pathogenicity and sort of the ability of this CAG repeat to expand has been shown recently to really rely on these slip DNA structures, which you can imagine here. And it's been known for a while, several decades, that these slip DNA structures preferentially form at larger repeats. So you have a longer repeat, you're more likely to form these slip DNA structures and more likely to have the somatic expansion, which has been sort of hypothesized recently to potentially be involved in the pathogenesis in and of itself of HTT in neurons. However, just thinking about this, going back to the initial slide where I said DNA doesn't exist, naked inside of the cell, but actually exists in the chromatin context, it's kind of challenging to imagine what's going on, how you can actually form these slip DNA structures if you're actually bound by a nucleosome. So our real question became is, when you actually have these repeat expansions, are you actually just occupying them by nucleosomes all the time, or are you actually preferentially keeping them in this sort of accessible or exposed, which may actually make them more inclined to undergo these repeat expansions? So to do this, we actually developed the targeted FibroSeq method. So I'm not going to go over the details of this, but really the major goal of this was to enable us to pool multiple samples into the same sequencing smart cell simultaneously, so we can actually go through higher throughput and get actually greater coverage out of the sequencing data. So we applied this initially to a quad where you had an affected father with Huntington's disease, unaffected wife, and then you had 2 children, one of which actually had juvenile onset Huntington's disease. So when you do this, this enables you to actually quantify initially, as you would imagine, the actual repeat length of the HTT CAG repeat. And you can see that in the affected father here, you have two different alleles, the expanded and the unexpanded, and similarly in the unaffected mother, you can appreciate the two unexpanded alleles. When you look at the children, you can see in this at-risk individual, you have the expanded allele as well as the unexpanded, and then this individual with the juvenile onset, you can see that this expanded allele even further expanded, and you're actually appreciating some of the somatic instability of that allele right here. So now you can ask, well, what's actually going on inside of the repeat? So inside of that CAG repeat, which is just a bunch of CAGs in a row, where you'd never be able to try to map short read data to this, you can actually now appreciate what's going on inside of that from a chromatin perspective. So what I'm showing you here is like, say, for example, on this fiber, you have this promoter right here, this +1 nucleosome, a little tiny internucleosomal linker region, another nucleosome that's actually embedded within this expanded CAG repeat, and then this large patch of accessibility that then transitions into another nucleosome out here. So you can see this on a per molecule basis now, what the actual chromatin architecture is inside this repeat. And if you actually quantify this and actually ask, well, how big is this accessible patch within, say, unexpanded repeats, or those which are expanded, you can appreciate that when you actually expand, not only are you expanding the CAG size, but you're also expanding the ability and the propensity for this to form a large accessible patch of chromatin. And not only, but going back to this diagram earlier, this is now in the size that's actually permissive for undergoing these slipped DNA structures. So what you have here is actually this ability where somehow how the HTT-CAG repeat was placed enables it to form this large patch of accessibility. We've repeated this on actually brain samples from individuals with Huntington's and shown this is not just in these sort of cell line systems of individuals with Huntington's disease, but also actually in brain samples in and of itself. Oddly, actually, this is kind of the interesting thing about this, other CAG repeats in the genome don't show this. So this is something unique to the actual HTT-CAG repeat, and I think it has a lot to do with this architecture here. And what I mean by that is you have this promoter, which is essentially creating this well-positioned +1 nucleosome, and the CAG repeat happens to be right here within this area between the +1 and +2 nucleosome. And that just happens to sort of be bad luck in terms of where this is positioned that is creating this large piece of accessibility. Okay, so to wrap up, really sort of some of the take-home points is that, functional data sharing, diverse human populations are really essential for us to move beyond sort of this VUS problem and actually get into sort of a better care for patients who are currently being given VUSs as a diagnosis, unfortunately. And really, I hope that the latter part of this talk has really shown you that some recent advances are enabling us to not just study the exome, but actually move outside the exome to better understand what's actually going on in terms of the genome, not only in the euchromatic areas, but also in some of these more challenging to map portions of the human genome. And similarly, you can now move beyond sort of a view of DNA like this when you're actually doing sequencing to actually what's going on top of that DNA at the exact same time. So you don't have to sort of double sequence your samples to get the DNA plus the epigenome, but actually you're able to do this simultaneously in the same reaction. So, and then I think that, the next steps for this is, how do we translate sort of what's fun technology and sort of fun on the research side into actual improved patient care? And I think that's really one of the big challenges that, I and others are, undertaking, over the next couple of years. And, one of the reasons why I'm really excited to sort of be here at the UW to sort of, take on this problem head on. So, as you can imagine, this was definitely not work that I could have ever done by myself, and this was, a huge team effort by many people to really enable this to happen. Wonderful people in the lab, Stephanie, who did a lot of the targeted stuff, Andrew Folta for the BBI, Ben as well doing the targeted. Itze has been really sort of wonderful with the protein preps. Shane and Adriana have been great in terms of the computational. Mitchell has done a lot in terms of the machine learning approaches, which I described. Elliot's done a lot of the work in terms of the de novo genome assembly, and Jane has sort of been essential for absolutely everything in terms of the wet lab, and Taylor similarly with the segmental duplication. And then other individuals, Danilo for the centromere story as well, and then many of our collaborators who've really just been essential for getting this off and running, and then obviously the funding, because none of this would be possible without it. So once again, thank you very much, and I appreciate any questions you have. So, yeah. Yeah.
Do I need a mic?
I can repeat the question. So.
Great talk, Andrew. It's fantastic work. I love how you talked about both the clinical and the cutting edge side. So I have two questions. One for your basic science, and then one on the clinical side. So the first question is, you talked at the beginning about your fiber-seq technology and showing heterogeneity molecule to molecule. So my question is, how did you rule out that isn't just heterogeneity of the enzyme you're using to do the methylation tag?
Yeah, great question. So this is actually one of the differences that we sort of initially were taking on when we were developing this technology. So typically when you're doing, say, a TAC-seq or a DNase 1, what you do is you add a whiff of the enzyme, like a tiniest amount, where it's just causing the selective cleavage within the accessible sites. Because what will happen if you add more, or sorry, the question is, how do we know the heterogeneity is, biological and not technical? But the reason why you do this with DNase 1 and TAC-seq is if you add a little bit more, what will happen is those enzymes will just cut in these internucleosomal linker regions, but when you start doing short-read sequencing, that just contributes to noise. So when we were developing this method, we ended up actually doing a lot of tests where we added that small amount of enzyme, then we added 10 times more, 10 times more, 10 times more, 10 times more. So actually right now we're adding around 1,000 times more enzyme than what you would do for, say, a DNase 1 experiment. And what we know is that we've actually sort of maxed out of this. So we can say right now, typically we're using 100 to 200 units of the enzyme. If we had actually 2,000 units of it, it doesn't actually sort of change these heterogeneity. Because essentially you're adding more enzyme there, but the nucleosomes are still occluding the ability of that enzyme to methylate the underlying DNA template. So, but yeah, that's sort of a difference in terms of the ability, because that additional cleavage with DNase 1 and a TAC-seq is noise, but for us that's actually signal. We like that cleavage or that methylation in the internucleosomal linker regions because now we can visualize sort of the presence of a nucleosome in addition to the presence of the accessible site.
And I'll point you to an ancient paper that was written by Stephen Tapscott and Hal Weintraub on single molecule stochasticity of gene expression. And are you familiar with that? You're nodding.
So, yeah. There's beautiful stuff like, from the 80s in terms of this as well. And also Stephen Tapscott has a beautiful paper looking at actually these repeat expansions, showing what's going on inside of it when it comes to the repeat expansion associated with DMPK, which is myotonic dystrophy type 1. But yeah, no.
You have the first data to actually experimentally support what they theorized and others. So that's super cool. My clinical question is more provocative. This whole problem of variants of uncertain significance, is this a problem that we, the lab, has created? Why are we reporting so many variants of uncertain significance? Why did we decide that was a standard clinical thing to do?
It's a great question, and one which I've thought about a lot in the past. I think that it all depends upon the user. And I think that's, this is also happening in terms of the labs. If you go to a prenatal testing, you're not reporting VUSs. You're only reporting likely path and pathogenic for carrier testing. That's sort of the standard of care across most labs. Now when we're talking about individuals who have a condition, then you actually add in this VUS. And I think it comes down to what level of uncertainty are you okay with. When it comes to carrier testing, you're less comfortable with giving VUSs typically because of the fact that most of those are going to be likely benign or benign. It takes 2 hits, because this is sort of in the situation of a carrier testing. So you feel more comfortable that you're not going to miss something out. But oftentimes when you're thinking about individuals who are coming to clinic with an actual phenotype and that you're trying to solve what's going on, it may be more uncomfortable to sort of say, well, there could be something here. There's not. But then it also comes down to provider comfort with this. The traditional model of thinking about VUS is where you're having a medical geneticist or someone with a genetics background who's actually doing the return of results, who may understand what that VUS means, how to best counsel it on it. And I think that one of the very exciting things over the last couple of years is that model is breaking down, and that we're seeing genetic testing not done by just genetic people with sort of a genetic background, but actually seeing more genetic testing done throughout the community of providers out there, which I think is great because that's the only way that's really going to improve access for it. And I think that as you're doing more of that, it's important to sort of ask that exact question that you just had, which is this traditional model of VUSs still workable and tenable? But the other challenge then becomes is, what do you do when it comes reclassification time? when you have a reclassification of VUS to a likely pathogenic and pathogenic variant, which is going to happen in probably 1 of 4 times, where it's going to be up-classified rather than down-classified. And are you then going to go from a testing report that said no variants identified to suddenly there was a variant identified? And does that actually reduce some trust that the patient had in terms of that initial test? Or is that actually make people more comfortable with the testing because they didn't have to sort of spend those years in this sort of VUS purgatory? I think these are wonderful questions. I don't have a great answer for it. I'm obviously coming in from a background of a medical geneticist where I'm like, I love VUSs, I'm fine with those. Like, I can deal with those because that's sort of our wheelhouse. Like, and I'm happy to sort of counsel patients on that and sort of advocate for trying to get these changed. So I would like to see them in reports, but I think that I'm speaking from a very narrow focus, and I worry that I may not be representing every single clinician out there who's going to be the future of genetic testing, which is not by medical geneticists. So,
how did you decide that it was a transcription factor binding site and not just a remodeling of the chromatin?
In terms of where?
Like you had a little green patch, like, oh look, there's a transcription factor binding.
Oh yeah. So you're talking about in terms of the CTCF binding element? So yeah, this is a great question in terms of how we're actually ascribing function to the footprints we're seeing. So I think that's, at the end of the day, when we do sort of fiber-seq methods, we're seeing, either a methylation or not a methylation. So it comes down to when I have a stretch of DNA that was unmethylated, am I surprised that it was unmethylated? Could it just be that I'm not adding enough enzyme or something like that? And also, what is doing that occupancy there to prevent the methylation from actually occurring? And so our first order of business that we typically do is to identify these large footprints that are essentially in the size of nucleosomes. So that's sort of like when we think about this from a computational perspective, we take our data and that's our first pass through it. Which usually these are peak at around 147 to 149 base pairs in length, which is sort of what you'd expect based on the crystal structure of nucleosomes. Then inside of these accessible patches, you have to ask the question, is great, I saw a stretch of unmethylated adenines. Am I surprised to see that? And what could be occupying it? And for that, there's a couple of different approaches that we've taken to address this. So one, in terms of your question regarding could this be a chromatin modifying enzyme or some other sort of polymerase binding there? To address that question head on, what we've done is actually sort of say, can we take this system and perturb it and see that what we perturbed is actually correctly changing? So if we knock down or sort of disrupted the polymerase, do we see that the footprints, what I'm assuming are polymerases, suddenly disappear? Or they increase if I'm sort of using a particular perturbational system that actually should increase the occupancy of that on the DNA. So that's been one approach we've been doing. The second approach is a little bit, so you can almost imagine more intuitive, which is to say, we already have a wealth of data regarding bulk occupancy patterns. So in that situation of the CTCF example, which I showed, we have ChIP-seq data to show that CTCF's bound at that. We have DNase 1 footprinting data on that same cell type to show that there's a footprint right there that likely corresponds to CTCF. So we can take this bulk data and now start asking the question of, great, we know that something's bound there in bulk, but what is the actual dynamics of that on a per molecule basis? And we've been sort of using a combination of both these approaches, but really I think this is sort of the challenge that we initially faced when dealing with this data is it's incredibly rich data when it comes to all this information. And now it's sort of pulling out these individual features that are actually modulating the methylation on a per molecule basis, where the challenge has sort of arisen. Yeah.
Okay, so you've established you can look at a per molecule basis at chromosome accessibility. Have you applied that to characterizing variants that may alter that chromatin accessibility and be able to classify those variants because you can see them on an individual molecule?
Yeah, so we're definitely doing that, and we are doing those exact same approaches. We're using sort of the targeted fiber-seq approach, which I described earlier, to sort of apply this to pathogenic variants or putative pathogenic non-coding variants to understand sort of how that's disrupting the chromatin architecture. Sometimes we know that for some of these variants, it's causing an effect on the gene expression, but we have no idea why. We have no idea how. And so using this per molecule data not only gets us sort of this question of, is there something distinct between the two haplotypes, the haplotype with the variant and the one with sort of the reference sequence there, but also what is distinct on the chromatin level? Is it the situation where actually that variant is causing the nucleosome to be better positioned, that's turning off the regulatory element? Or is it disrupting a particular transcription factor binding element? You name it. So we can start to piece those things together with this. So yeah, we have a bunch of, several projects actually focused on that specific question. So yeah. If you have any good cases, I'd be happy to do that as well. So.
Well, I was just going to say, I think it'd be interesting to know just what is the diversity between the two haplotypes, right?
So that's actually one of the big, so the challenge with all this stuff is depth. So like, with this heterogeneity, how much depth do we need to actually get? So we're actually just getting our first data back where we have now around 100 fibers at each given spot along the genome. So it's 100x coverage of the entire genome with this. So now we can actually start looking at what is the expected amount of haplotype, or heterogeneity that we'd expect on a given haplotype. And now that we have this, can we start applying this across different tissues and actually understand what is this sort of expected across diverse haplotypes within a population? So, yeah, and some of the new sequencers which are coming online are really enabling this to be possible. So, yeah. Thank you very much. Yeah.